So this short lecture is about hearing. It's really about the biology of how we transduce sound waves into neural signals. So just a quick anatomy of the ear. We'll go through this again. But this is the outer ear, and this is the external auditory canal. There's the tympanic membrane, auditory ossicles, bones, and the inner ear of the cochlea. So sound waves are really a compression and rarefaction, a pushing together and pulling apart of air. Now we can have sound waves actually transfer through water and through metal, but we need some substance. You can't have sound, for example, in a vacuum or out in space. And that's why when you're watching movies and there's this big giant explosion in space, really there wasn't there might have been an explosion, but there was no sound produced from it. So these sound waves are going to be transferred into the tympanic membrane or the eardrum. Now, a few things about sound waves. The more waves that we have in a given amount of space, the higher frequency of waves in a given time, the higher the pitch, the higher the frequency of sound. So you have a very high tone, high frequency when you have a lot of waves. When the waves are stretched out a little bit more, you have a low tone, right? A low pitch. This will become important when we look at the mechanisms of transduction and a little bit about perception. So the air comes through here, hits this very sensitive membrane called the tympanic membrane or the eardrum. This air pushes these bones, the malleus, the incus, and the stapes, these bones, and you go from a larger surface of the tympanic membrane and focuses that energy into a smaller surface called the oval window. So that focuses the energy because we're going to have to transfer that sound in air into the same waves in water or a fluid that is inside the cochlea. So we need to focus that energy. So those are the auditory ossicles. The stapes here is going to hit that oval window. It's going to push that membrane, push fluid on the other side. Malleus, the incus, and the stapes. These are going to really magnify that energy. And finally, in here is where we're going to spend a lot of the lecture, and that's really talking about where we get a transduction. So we've gone from air waves, sound waves in the air, to water waves. And those water waves are going to flow really through this area here called the cochlea. And we'll take a look at the cochlea now. So if I was to slice the cochlea, slice it, or unwind it and slice it, because it's really wound up like a snail shell, you're going to see that there are three chambers. These are fluid-filled chambers. And right in the middle chamber here, you have an area called the organ of Corti. And the organ of Corti sits on a membrane called the basilar membrane, which we'll talk about in a few slides, which bounces up and down, up and down. And then you have this rather stiff membrane at the top called the tectoral membrane. And between them, you have nerve cells. You have nerve cells that send signals. And what's going to happen is, as you push these nerve cells, also called hair cells, into the tectoral membrane, that's going to create a mechanical shift in ion channels. We're going to open up ion channels. We'll talk about that here in just a moment. I want you to get an idea that the organ of Corti is just not one thing sitting here. It really stretches all the way along the whole canal. Okay? And remember that these are fluid-filled spaces. And the oval window is going to have a lot to do with how the waves, water waves, fluid waves, go through. So the basilar membrane, where the organ of Corti sits, is between the cochlear duct and the tympanic canal. It's tough but flexible membrane that moves itself in the direct response to the frequency of the incoming sound, and that's very important. So the frequency of the waves in the air are going to transfer into the frequency of the waves in the fluid inside the cochlea. So sound waves turn into fluid waves. It's much stiffer near the stapes, near the oval window, and it gets more flexible as we move out, as we move down the basilar membrane. So let's look at it again. Basilar membrane, we're just slicing it. This stretches all the way back. This stretches all the way back. And there is this fluid in here. And there's lots of these hair cells. And everywhere along these hair cells, you're going to be connecting to the auditory nerve. I think it's actually called the vestibular cochlear nerve now. Okay, so the organ of Corti, very important, is the place for transduction. The place where we're going to convert something in the world, in this case a sound wave, into something the brain can deal with, into the code of the brain. And the codes of the brain are nerve signals, action potentials. The organ of Corti contains a lot of hair cells, the inner hair cells, the outer hair cells. And these hair cells are connected with a filament to the tectoral membrane, the top membrane. Let's take a look at that. Okay, so there are the fluid-filled chambers. Here's the tectoral membrane. Here's the basilar membrane, moves up and down. Basilar membrane moves up and down. Here are the hair cells. Let's magnify that a bit. And you can see these are neurons. These hair cells are neurons. And they're firing, the rate of firing has a lot to do with the polarity of the membrane, like everything else. But how does that polarity change? How do the polarity, the inside of the neuron compared to the out, how is it changed? Well, it's changed by ion channels, just like all the other neurons we've talked about. How are those ion channels affected? And here's where we get something a little different. They're not affected by a chemical, like a neurotransmitter. They're not affected by electricity, like the voltage-gated ion channels. They're affected by a mechanical process, by the stereocilia opening up and closing these ion channels. As the basilar membrane pushes up into the tectoral membrane, these are going to be pushing back and forth. And almost like a lever, think of it like a lever opening up, like a lever opening up these ion channels. So if the wave happens to be hitting at this spot along the basilar membrane, remember it's a very long thing, if the wave is really pushing up right at this spot and not other places, we're going to get a lot of pushing against the tectoral membrane. We're going to open up a lot of ion channels. We're going to change the polarity. At least this area knows whether a wave is hitting it or not by the amount of mechanoreceptor activity, the amount of opening and closing of these ion channels. So here's the basilar membrane all stretched out. Organ of Corti is sitting on it. Here we are down by the oval window, and here we are way at the end here. Now, what's interesting about the basilar membrane, which we'll talk about now, is that the waves are hitting it at different spots. If we have a very deep wave, very long wave, it's going to hit down here. But if we have a high frequency wave, that high frequency wave is going to hit right here. So the basilar membrane becomes very sensitive to different frequencies of waves. Lower waves down here, higher waves up here. So this goes into one theory I'd like you to know. It's a theory put together by a guy named Georg von Bekesy. He won a Nobel Prize for this work. And he said that the organ of Corti along the basilar membrane is tonotopic. That means it's a map of tones. Hair cells of the organ of Corti are organized in a strictly tonotopic fashion. High frequency hits near the oval window, and lower frequencies hit further along. There's a picture of Georg von Bekesy. I actually worked at the Bekesy Laboratory of Neurobiology in Hawaii because he was a professor there for a long time. Okay, here's that basilar membrane. High frequency here, low frequencies down there. Okay, so all the way along the auditory, the nerve, all the way along the basilar membrane, we have the auditory nerve being sensitive to different frequencies. For example, way at the end, the auditory nerve is sending a signal if it's being pressed that there's a low frequency, really low, down, I don't know, maybe 50 hertz, 50 cycles per second. The area of the basilar membrane and the auditory nerve sending signals near the oval window at the beginning are very sensitive to high frequencies because those frequencies are hitting there much sooner. High frequencies, maybe 15,000, 18,000, those are the highest frequencies that really humans can hear. The younger you are, the higher the frequency you're able to hear. As you get older, you start losing your higher frequencies. So that is the tonotopic nature of the auditory nerve. Okay, so there's just another picture of it. Okay, higher frequencies down, higher frequencies up here, lower frequencies over here. This is where the fluid is being pushed, and if it's a low frequency, it's displacing here. If it's a high frequency, it's going to peak much sooner. Okay, so the auditory nerve, or the vestibular cochlear nerve, is going to send a signal to a lot of places in the brain. And it's going to send signals both contralateral and ipsilateral, but it's going to end up at the thalamus. It's not going to hit the lateral geniculate nucleus of the thalamus. It's going to hit the medial geniculate nucleus of the thalamus, and eventually head on to the auditory cortex. The auditory cortex is also tonotopic. In other words, the high frequency area of the auditory cortex is getting signals from near the oval window. Those nerves are sending their signals via the medial geniculate to here. A little lower, a little lower, a little lower. And so the auditory cortex, like the basilar membrane, is tonotopic. And from there, we're going to start decoding that information, that very complicated information, because sound is very complicated. And so and over here, you're going to start getting into areas that start decoding language and start decoding the very specific memories of sound.